The Origin of the Genetic Code, a scientific examination by Thomas Seiler, Stuttgart, Germany. A typical computer contains a hard disk drive, which uses a code language based on a binary alphabet, a sequence of zeros and ones. This code system achieves a data storage capacity of 1 gigabyte per cubic millimeter. That means one can write the information content of 1000 medium-sized novels on a tiny cube with a side length of 1 millimeter. The natural genetic code that can be found in the cells of every living creature on Earth uses a quaternary alphabet composed of 4 symbolic letters A, C, G and T. A sequence of 3 of them, a triplet, represents a word. The sequence of many such triplets corresponds to a sentence. The material of which the letters are made and with which they are bound together is called deoxyribonucleic acid, abbreviated as DNA. The data storage capacity of this code system is 1 billion times higher than that of the state-of-the-art computer hard disk drives. The computer code system was developed by very many scientists, applying intelligent research and design work. The much superior DNA code has not been developed by many human engineers. The following slides will examine the question: is it possible that it has been produced by blind natural processes? Deoxyribonucleic acid, DNA, is the long molecule that carries genetic information for the development and functioning of an organism. DNA is made of 2 strands that wind around each other like a twisted ladder, a shape called double helix. Each strand has a backbone composed of alternating sugar, deoxyribose, and phosphate groups. Connected to each sugar is one of the 4 nucleobases: adenine, cytosine, guanine, or thymine. The sequence of the 4 bases along DNA's backbone is a code, which contains biological information, such as the instructions for the assembly of a protein or an RNA molecule. A code-carrying DNA molecule is entirely ineffective and useless if it is left on its own. Only in combination with a complex reading and construction machine is it possible to make use of the genetic code stored in the DNA molecule. Such a machine is present in the cells of biological species and is called ribosome. A ribosome reads the sentences written in copies of the DNA strands and uses their coded instructions to produce amino acids, another sort of complex molecules, and arranges them as long sequences with a specific geometric folding structure. These macromolecules are called proteins and carry out biological functions in the cells. The question whether the genetic code can have arisen by natural processes is therefore always combined with the question whether the ribosome machinery can have arisen by natural processes. Without the latter, the first would not make any sense and therefore not exist. Before continuing with the examination what nature is capable of producing, it is helpful to consider the results of intelligent man-made chemical engineering. Scientists have tried to produce DNA molecules starting from inorganic molecules like hydrogen, carbon dioxide or methane, elements that are supposed to have been present in a primordial earthly environment before biological species have existed. Using sophisticated chemical reaction sequences with carefully chosen parameters like temperatures, pH values, reaction times, ingredients, purification procedures, and so on, they succeeded in producing some of the organic molecules like deoxyribose, phosphate groups, and nitrogen-containing nucleobases that represent building blocks of the long DNA macromolecules. Using additional chemical ingenuity, they even managed that these building blocks stick together as a chain of a polymer. Copying the arrangement of natural DNA found in living cells, they could even produce synthetic DNA polymers that contained a sensible code. What is the bottom line of all the research efforts dedicated to this? First, the results, or better, the ways on which these results could only be achieved, underlined that much intelligent engineering by a skillful scientist is needed to produce organic molecules in usable, purified forms and connect them to sensible DNA. This is in contrast to all known natural processes. Even in a primordial soup scenario, they are only capable of producing tiny amounts of organic molecules, like nucleobases or amino acids, that are accompanied by many side products. These side products are present in large quantities and inevitably lead to the decomposition of the unstable organic molecules and effectively block their binding to form macromolecules like DNA polymers. Furthermore, no natural process is known which would be able to write a sensible genetic code. An intelligent mind is required that can purposefully arrange the nucleobases in meaningful sequences. In an article in Interdisciplinary Science Reviews, Klaus Dose summarized the difficulties for an unguided, naturally occurring chemical process to produce the basic building blocks of living cells. In an aqueous solution, the reactive intermediates of the Miller-type experiments would readily interact to yield more complex and ill-defined materials, but practically no biologically relevant molecules. It is extremely unlikely that the first forms of life could have evolved spontaneously in a primordial soup. In the journal The Guardian, Paul Davies pointed out that we cannot obtain useful DNA strands by simply producing their chemical building blocks. DNA is not a special life-giving molecule, but a genetic data bank that transmits its information using a mathematical code. Most of the workings of the cell are best described not in terms of material stuff, hardware, but as information or software. Trying to make life by mixing chemicals in a test tube is like soldering switches and wires in an attempt to produce Windows 98. It won't work, because it addresses the problem at the wrong conceptual level. And Hubert Jockey explained in the Journal of Theoretical Biology that there is a fundamental systematic barrier preventing a gradual evolution of a biological data storage device from simple organic molecules. The logical reason is that a storage device can only record a certain information content if that device itself has already got this information content before it is able to record it, and thus pass it on to the next generation. He said, "There is no gradual sequence of events from the primeval soup to an element that records the degree of complexity, that is, information content, from which the process could grow exponentially. That degree of complexity, measured in bytes, must be had in order to begin." This leads us to the question why we cannot observe natural processes that produce ordered constructions like that of a DNA double helix or of a protein. The answer is found in the most basic natural law that governs all physical and chemical processes in nature, the so-called second law of thermodynamics, which is just a description of our all-day experience in scientific terminology. The textbook formulation says, "A system will never change by itself into a significantly less probable state. That means its entropy will never decrease by more than a few K."
Hello, this is Rob Stadler, and I'm here to present a 20-minute opening argument on the topic of the impossibility of the origin of life from non-life through purely natural or material processes. Sometimes this is known as abiogenesis. The basics part of my thesis here is that the more we learn about the requirements for life, even the simplest known forms of life, and the more we understand about physics and chemistry and thermodynamics, in other words, science, the more that the science that we understand supports the conclusion that life could not possibly have started through purely natural processes. And I'm going to explain this in two directions. The first will be a bottom-up approach, basically going from basic chemicals, very simple molecules like methane, carbon dioxide, ammonia, water, and how the struggle would be there to develop from those basic chemicals into something that we would call life. And later I'll take a top-down approach, basically explaining all that we know about life and the simplest possible forms of life and what it would take, sort of setting the bar there for basic chemicals to try to get to that point of life and the challenges there. So first we'll go with the bottom-up approach. And I have actually 12 points to raise there. There certainly are more points, but I picked my top 12. So let's start with very basic chemicals. And I'll tell you that what we see in life, what we know from science about what is in life, is that life is based on rather a small subset of select molecules, basic building blocks. And that would be something like 22 types of amino acids, very specific subselection of the possible types of amino acids that could be produced. Life only looks at 22 of those. We also have a subset of nucleotides of 5 particular types of nucleotides that make up DNA and RNA. We have select kind of phospholipids in life, select kinds of sugars. So life is very picky about the kinds of building blocks that it uses. On the flip side, from science, from physics, from chemistry, we know that if you take basic molecules, like I said, ammonia, water, methane, carbon dioxide, and you mix them together and they start to react with each other under various conditions, what you end up with is a very, very diverse mess of millions of different types of resultant products from that. And it therefore becomes very difficult to sort out only those that you want for life, only those relevant to life, from the mess that the random natural process creates. And those millions of types of diversity of molecules actually get in the way of trying to form a living thing or trying to get molecules together to make polymers steps toward life. And they can also sometimes be toxic, literally breaking down or destroying the molecules that you do want. So natural processes oppose what we want to see for basic building blocks of life. And this we know experimentally from examples like the Murchison meteorite, which contains thousands, tens of thousands, millions of diversity of small types of molecules, and only a small, very small subset, very dilute, of those that are related to life. We also know from a computer simulation of chemical reactions known as alchemy, that was published in Science a few years ago, that shows that you get a vast predominance of types of molecules that you don't want for life. when you mix things together. Second point is homochirality. Most organic molecules, lots of organic molecules, have a property known as chirality, where the same exact molecule can have a right-handed form and a left-handed form that are not superimposable. Life is very selective about that. Life is exquisitely homochiral, meaning that only left-handed amino acids are used, and what's generally considered right-handed nucleotides are used in life. Now, we also know that natural processes do not produce that except in extremely rare situations. Natural processes produce racemic mixtures, which is an equal 50-50 mixture of left-handed and right-handed products because they're thermodynamically equivalent. So we know what natural processes do, and we know what life has, which is very distinct from natural processes. The third topic is that we know that life, all of life we know, is based upon water. Water is the basis of life. But life also requires polymers, such as proteins, such as nucleic acids, that are formed from monomers coming together, perfectly lined up in a long sequence. And those monomers, when they join together to make the polymer, they come together through what's called a condensation reaction, which actually produces water as the polymers formed from the monomers. So we know from science that these condensation reactions are not favored in water. In fact, they're opposed. They're naturally opposed when you're in water because of Le Chatelier's principle. So some try to get around that, let's say, by doing wet-dry cycles, for example, wet and dry, to try to get these monomers to form into polymers, even though it's a condensation reaction. However, we also know that proteins are denatured by drying out. They lose their shape, and then you put water back in, put them back in solution, and you no longer have a useful shape. Proteins are very dependent on their shape in order to be useful. So that's a problem. And it's very unnatural to have life be based on water and yet requires polymers that are formed by condensation reactions. Another topic, number 4, is that these polymers, proteins or ribonucleotides and DNA, they always have consistent bonding, which we would call a 3' 5' phosphodiester bond. They're perfectly linked up, kind of like cars on a train track full of boxcars, perfectly lined up every time. Whereas natural processes will tend to link monomers in all kinds of different ways and end up with the wrong kinds of bonds. That's just what naturally happens. So it's very unnatural to have a perfectly lined up polymer of any kind of length. And of course, the polymers we speak of in life are extremely long and extremely precise in their kinds of bonds. The next item is that these information-containing molecules such as DNA and RNA in life must be maintained. They don't just sit around forever. They naturally degrade. Natural processes attack molecules like RNA, even simple water, high pH, divalent magnesium, which are very common, are going to degrade the RNA very quickly over time. It'll just fall apart. So we know that every kind of life form has to put out a lot of energy and have very sophisticated machinery in order to maintain those information-containing molecules, or else the information is lost over time in what's called error catastrophe. Basically, everything falls apart. Next, we know that these information-containing molecules, like DNA and RNA, have to be replicated. That's a requirement for life. You absolutely have to replicate. For example, DNA replication in its simplest form requires about 25 specific proteins that come together to form 14 enzymes, molecular machines that are specifically there for the purpose of helping to replicate the DNA. It's extremely complicated to come up with those 14 enzymes. Natural processes, on the other hand, do not replicate molecules. We know of no self-replication of any information-containing molecule that has ever been observed or demonstrated. The latest results trying to show this from Gerald Joyce's lab, he published a few years ago, and he claimed that he had a ribozyme, an RNA enzyme, that could replicate its own ancestor. But if you read that paper, you find out that that's quite a stretch of a conclusion, because this ribozyme at best could only replicate 1/3 of pieces, 3/3 of its former self. And the yield was so poor for those 3 pieces that the scientists had to step in, intervene, and get rid of all of the junk that it produced, leaving only the good stuff, so that those could combine to replicate its ancestor. And then they found, after doing all that the fidelity of the replication was only about 92%, which is completely insufficient to maintain information over several generations. Like I said, you end up with error catastrophe. So nothing even close has ever been shown for this needed essential goal of self-replication of an information-containing molecule. Natural processes simply oppose what is essential for life. Next point is that any kind of life, there's a lot of junk that's produced by life, mistakes that are made, broken machinery or machinery that produces faulty parts. This is replete throughout life, and life can't exist without a means of identifying and recycling or removing that junk. There is no known mechanism in a purely natural process that is smart enough, specific enough to identify bad product or bad machinery and recycle it or remove it. So that also goes completely against natural processes. We also know that life, of course, is based on metabolism, and that metabolism, keeping a living thing alive, is always driving that life into a state that is far from thermodynamic equilibrium. It has very low entropy, meaning it's highly organized, highly structured, and very high enthalpy. And that is a very unnatural state to be in, because we know that spontaneous chemical reactions are always going to be favored, those that have negative Gibbs free energy. Thermodynamically, you have to have negative Gibbs free energy to have a spontaneous reaction occur. That's natural. What we see in life is the opposite of reactions being driven toward far away from equilibrium, because they have such low entropy and high enthalpy. Next, we have in life, we observe that you absolutely have to have homeostasis, which is a highly regulated, and it's maintained by a membrane that's active and selectively permeable, so that inside the membrane, inside the cell, you have homeostasis. You have a very consistent environment, despite very dramatic changes that can happen outside. Changes in temperature, changes in concentration of molecules, changes of pH, that can happen outside. But inside, you have a steady, consistent, maintained environment. Life requires that. Life does that. That doesn't happen naturally. You can't have that sort of, again, it's a being far from equilibrium state that is maintained with life. And we don't know how to do that through purely natural processes. Next, in life, we observe that there's a strong interdependence of a variety of types of molecules. Specifically, keep a short list here, that RNA, proteins, and DNA in every form of life that we know about are highly interdependent. What I mean by that is that you can't have proteins without RNA, which is used to produce the proteins. You can't have RNA without the DNA, which is used to produce the RNA. You can't replicate DNA without proteins and RNA that are there to replicate the DNA. So there's a huge interdependence between the ingredients, the essential ingredients of life. And this is kind of a summary of what's been said about natural processes, but natural processes do not or cannot ever produce a variety of homochiral, perfectly linked, purified biopolymers in one location that each cannot exist without dependence upon the others, that's just not a natural thing to see. And taking that a little bit further, we know that life, all of life, even the simplest life, is replete with examples of what we call circular causality, which is basically saying chicken and egg phenomenon. And I'll give you a simple example that all of life uses adenosine triphosphate, ATP, in order to harness energy. And that's kind of like a charged up battery. The discharged battery is known as adenosine diphosphate, ADP. Life first needs to produce the discharged batteries, ADP, and then charge them up to make ATP. However, we know in life that making one molecule of ADP, the discharge battery, requires 7 molecules of ATP. So you can't get ADP without ATP, and you certainly can't have ATP without ADP. Simple example of circular causality. And that's the kind of thing that natural processes do not do. They don't solve these chicken and egg problems. Intelligence can do that. Like we can produce cell phones, which are useless unless you have a cell phone tower. And we can produce cell phone towers, which are useless if you don't have a cell phone. But human intelligence can produce that together, that circularly causal system, put it all together and have it all work out. Another example in life is Eigen's paradox, where you can't have life without repair mechanisms for DNA and RNA, but those repair mechanisms are themselves stored within the DNA and RNA and require such a length of DNA and RNA that cannot be maintained without repair mechanisms. So that's another kind of causal circularity, and there are dozens of examples like that, which are highly unnatural. They don't just pop up through natural processes. Physics and chemistry working all on their own. And then one last example is the way that life harnesses energy. Life requires very specific forms of energy harnessed for particular purposes in its metabolism. Again, keeping it away from thermodynamic equilibrium, far away, so it's highly organized and in a high energy state. All of life uses ATP. All of life requires ATP synthase, an extremely complicated 20-protein enzyme that charges up ADP into ATP. It also requires a proton gradient. All of life requires a proton gradient. And all of this is for the purpose of harnessing energy, very carefully, very specifically using energy to drive it away from thermodynamic equilibrium. When we look at natural processes, we see sources of energy that are raw, like radiation, sunlight, UV light, volcanoes, lightning. Those kind of energy applied to molecules are predominantly destructive. They destroy, they rip things apart, they create a whole pile of garbage, but they certainly don't provide careful, organized, low entropy, high enthalpy, organized molecular systems that are driven far away from equilibrium. So that's just a short list then of all the ways that what we know about life is quite distinct from what we know about what natural processes, chemistry, physics on their own do. Now, just 2 minutes now on the top-down process that I mentioned. Scientists for years have been trying to find the simplest form of life and even intervening to make it simpler than that, as simple as they can get it, simple as they can. They want that just to understand what is the requirement of life. And Craig Venter's Institute has done this, and they keep pushing it. And they have created something called JVCI SYN3A, and the most simple known form of life. It's a derivative of mycoplasma. It has 493 genes. And that requires about 500,000 base pairs of DNA. So that's 500,000 letters of DNA. That's how much information is in the DNA in that very, very simple life form. And of course, we know that DNA is not the only form of information in life. It's just one particular form, but 500,000 letters. It has a membrane. The membrane, in order to maintain homeostasis, it contains more than 100 different kinds of proteins. And they are there to facilitate transport across the membrane and to maintain homeostasis. It requires special DNA repair mechanisms in that very simple form of life, JVCI SYN3A. It requires ATP synthase. Dozens of proteins are there to translate RNA into proteins. Our DNA gets transcribed into RNA, and RNA gets translated into protein. That requires about 100 genes, minimally, absolutely minimally. And this, although it is alive, this cell, it's very, very sensitive. It's very delicate, I would say, fragile. And it must be coddled, I would say, by the scientists keeping it alive, because they have to provide just the right nutrients, just the right time. And they have to remove its waste, giving it kind of a coddled existence, nurturing it to keep it alive. This is not the kind of life that could survive on a lifeless planet in the early stages of a prebiotic world, trying to keep it alive. It's very sensitive and couldn't do that. We're talking about an obligate fermenter, and fermenters actually don't exist alone in the wild unless some other form of life cleans up their waste. And sometimes that might be a scientist in a lab, but they can keep it alive only by special attention and coddling. So my point is that we know from studying life, even the simplest forms of life, that those very simple forms of life are far from being very simple, extremely complicated, loads and loads of requirements, loads and loads of required information, organization, layers of organization. And, that kind of sets the bar for where natural processes would have to build up to get to that bar before you would have anything close to what we would consider life. So that would be the summary of my thesis there, is that we know enough about thermodynamics, chemistry, science, physics, and we know enough about life at this point to say that with each passing year, it becomes more and more clear that natural processes on their own could not have started life. Thank you.
All right, my name is Pamela Acker, and I'm here with the Kolbe Center for the Study of Creation, and I'm going to talk today about genetic entropy and whether or not mutations can be a source of new information for evolution. So before I begin, let's start with a prayer. In the name of the Father, and the Son, and the Holy Ghost. Amen. Saint Michael the Archangel, defend us in battle. Be our protection against the wickedness and snares of the devil. May God rebuke him, we humbly pray. And do thou, O Prince of the heavenly host, by the power of God, cast into hell Satan and all the evil spirits who prowl throughout the world, seeking the ruin of souls. Our Lady, Queen of the Angels, pray for us. In the name of the Father, and the Son, and the Holy Ghost. Amen. All right, here we go. So just to give you a little bit of a background, I just put all my stats here on the screen. I'm not going to go through all of them in great detail, but I started working with genomes when I was an undergraduate at Southeast Missouri State University. I was hand annotating fungi genomes, which is just as boring as it sounds. But after that, I worked for a while at the Genome Sequencing Center at Washington University in St. Louis, where I was a whole genome library maker. So I took the samples that people gave from the species that they wanted to research. They sent in a sample of tissue or DNA. I extracted the DNA and broke it into little pieces and sent it along to the next stage of the process. So I didn't actually work on the Human Genome Project, but the Human Genome Project was underway while I was at WashU. So that was pretty cool. I went back for a PhD in biology in 2010, and I graduated with a master's in 2012, primarily because I refused to work with aborted fetal cells in the research that I was doing. So I was looking at developing an HIV vaccine, and my part of the project was ethical, but the project as a whole, I found out about halfway through employed aborted fetal cells, and I told my PI I couldn't do that. And so I ended up transferring to a different lab working with nematodes, but ultimately left with my master's degree because I was not interested in studying worms for the rest of forever. And in between jobs researching in the lab, I've done a lot of work explaining really complicated biology to people who are not biologists. So that's kind of my background and where I'm coming from. So I can keep that in mind as we go forward. So I want to talk first about mutations being a mechanism for evolution. So if evolution is to explain all of the diversity that we see in the world today and every living organism, how it came to be, it requires increasing genetic variation in a population in order for that to work. So evolution works through mutation and natural selection. You have new variations that arise in a population. These new variations are selected for or against. The next generation looks more like the variation that are more favorable. Okay, the only currently identifiable source of this new genetic variation in a population is mutation. Now, you can get variation in an individual through other means like homologous recombination or something like that, but the only way to get it in a population is through mutations in individual genomes. So since populations are what evolves, not individuals, no amount of homologous recombination is going to give us any new variation. It has to be mutation. So if mutations are necessary for evolution, and I can show you pretty conclusively that mutations don't actually do what evolution needs them to do, they're not a really good mechanism, then we have a very good reason to doubt that evolution is how everything came to be. So that's what I propose to do today. And we're going to start with an analogy because it's a little bit easier than going into all the technical details, but it still is going to make sense. Okay, so you can think of genetic information or a genome as an instruction manual that codes for how to build an organism. So if you have an instruction manual to build a giraffe, that instruction manual is going to contain thousands and thousands and thousands of pages of information because a giraffe is a very complicated organism. Now, giraffes are selfish, so each giraffe has to have his own instruction manual. He can't borrow from his neighbor or his mom or his dad or his brother or sister. He needs that information because it contains everything he needs to build his own body and keep it running. And since he needs his own instruction manual, he's got to copy it from somewhere. And since giraffes didn't invent a printing press, they each have to painstakingly copy the instruction manual from their parents by hand. And they copied it from their parents by hand, and they copied it from their parents by hand. And this is actually a pretty good analogy for how genetic information is passed along. It is copied in the cell, and that copying is done base by base. And you can think of bases in the genome as letters in the instruction manual. So whenever I say base, think of it as like a letter in the genetic code, okay? Now, there's a problem with copying instruction manuals by hand, whether you're doing it in the terms of DNA replication or you're doing it in terms of a giraffe using a scribe, okay? That problem is that you're going to introduce mutations or mistakes into the instruction manual every time you copy it. Now, each generation is going to introduce new mistakes. So if you were to copy a book, right, if I handed you a copy of a book that I'd made 5 mistakes in, you don't know that those are mistakes. So you're going to copy the book, you're going to copy my mistakes, and you're probably going to introduce a few of your own. And that's what happens every time something is, a generation passes down, right? So in human beings and in some other animals, it's about 100 mutations per generation. So my grandparents made 100 mistakes when they were copying the DNA that they gave to my parents, right? My parents made 100 of their own mistakes in addition to 100 of my grandparents' mistakes, and they passed those 200 mistakes down to me. If I make 100 of my own mistakes, I'm going to pass 300 mistakes from 3 generations on down to my offspring. So you see there's an accumulation of mistakes, accumulation of errors over time in the instruction manual. And as we have an increasingly large number of copy errors, we're headed towards not a new organism, not a giraffe evolving into, a giraffe with wings, or a giraffe evolving into something that can eat a different kind of food or prey on lions or something like that. What we're heading towards is a giraffe that is no longer going to work. What we're heading towards is total genome collapse. And that concept is genetic entropy. And that was a term coined by Dr. John Sanford, who's written a wonderful book about this and answers basically all of the questions that contemporary scientists raise either in the book or in subsequent articles that he has written that are available on creation.com. So if there's anything specific that gets brought up today that I'm not able to answer or my colleagues aren't able to answer, I would go to creation.com first and look up Sanford and genetic entropy, and he's got an answer for it there. And if not, he already answered it in his book, which unfortunately a lot of people haven't read. So he makes a very good case for how mutations actually lead to genome catastrophe rather than to evolution of new organisms. So I'm going to look at that, but I'm going to look at it kind of from the reverse angle. I'm going to look at what it would take for mutations to be an actual mechanism of evolution. All right. And fitting with my analogy, I've simplified this a little bit so that it makes sense. All right. So for mutations to be the mechanism of evolution, most of the instruction manual should be useless, because if it was mostly useful, every time I had a mistake, that's going to degrade information that I need, right? If it's mostly useless, it's no big deal if I have 100 mutations every time I make a new copy. Most of the changes, most of the mutations should be harmless, okay? Again, if most of it is junk, most of them will be harmless. No big deal, right? Some of the changes have to be good. They have to improve something. They have to give me a new function. A new way of walking, a new way of eating, a new way of a protein doing something in the body that's just a little bit better, right? And the accumulation of those changes in the instruction manual has to lead to a new kind of animal, right? I have to start with a horse and end up with a giraffe. That's the kind of thing we're talking about here, okay? So I'm going to look at each one of these individual points and talk about research that's been published in peer-reviewed journals and why each of them doesn't hold water. And if I can junk any one of these, I've junked the mutations as mechanism of evolution hypothesis. But just to be thorough, I'm going to junk all 4 of them. So here we go. All right. The first problem is that there isn't useless DNA. So after the human genome was sequenced, there was a project started called ENCODE. And ENCODE hadn't been going on for very long before they recognized that actually the majority, if not all, of human DNA is actually functional. Now, how did they figure this out? Well, they looked at 2 things, okay? And these 2 things are understood by all people who support evolutionary biology as being indicative of function. So this is not a creationist point of view. This is a mainstream evolutionary biologist point of view, okay? The 2 things that I'm going to talk about today, that most of this DNA is transcribed, and I'll explain what that means in just a second. And a surprising amount of it is conserved, and I'll explain what that means in just a second too. Okay. So the central dogma of molecular biology, there's more text on this slide than I need, but the text that we need is bold, okay? Is this idea that the DNA is transcribed into RNA and the RNA is translated into protein. This shows you the information flow from the DNA to the protein. And the protein is the stuff that you need. It's the stuff that your cell builds stuff out of and makes stuff with. Without protein, your cell wouldn't do anything functional. It would just kind of cease to be, would not be useful at all. Okay. So proteins are super, super, super important. And most of what DNA does is tell your cell what proteins to build, how to build them, when to build them, where to build them, and not really why, because it's a cell, okay? But that process of when and what gets put together in a cell and produced in a cell is extremely important. And we'll come back to that. We'll touch on that again a couple of times throughout this presentation, okay? So keep that in mind. All right. So your DNA, think of your DNA, since it is the instruction manual, it's the only one the cell has, right? It's sort of like a rare book in a library. You wouldn't give a rare book in a library to a high school student if you wanted to use a little bit of it for a research paper, right? You'd make a photocopy and you'd give that photocopy to the high school student, and then if it gets damaged, it's no big deal. So the DNA, the copy of the DNA that's made is called RNA. So when we say DNA is transcribed into RNA, what we mean is that the information is being copied from the master copy into a working copy. If I make a working copy of the DNA, that is evidence that that DNA is functional. Because if I did that and it wasn't functional, that would be a huge waste of energy and resources. That would be very disadvantageous, and it should be selected against if evolution were correct. Okay, so this would simply not be happening in cells if all of that information were useless. Right. The other thing that indicates that a lot of this information is useful is there's a lot of sequence conservation. So if you look at the DNA sequences of individual organisms, and I'm showing you here a figure from a paper that was in the Corsi lab where I worked at when I was at Catholic University of America. This was the second lab I worked in. We were looking at gene sequences in nematodes. And so someone had aligned these 5 different species of nematodes. And the reason those green boxes are PA1 and PA2 is because those are the ones that I picked out that we should look at and work on and study what happened when we mutated these areas. Because since those black shaded boxes indicate that sequence is conserved across all 5 of those species, that's telling me that is a very important sequence. That is a necessary sequence for these organisms to grow and develop and do what they need to be doing. And that is definitely going to be a good place to look for functionality in the genome. Okay. Now, sometimes evolutionists want to argue that, well, sequence conservation indicates that we have a common ancestor, right? But humans have sequences that are 100% conserved when you compare them to mice. Yet no evolutionary biologist thinks that humans and mice share a close and common ancestor that would lead to those sequence conservations. So they will say those sequence conservations are related to function. Now, I would say most sequence conservation, if not all sequence conservation, is related to function. So evolutionary biologists already admit that when you see sequences line up like this, it's evidence that they have function. So we have 2 major points of evidence that most human DNA is functional, right? Evolutionary biologists sometimes, though, will throw up things like introns or pseudogenes and say, well, these are evidence that there's a lot of junk DNA in the genome. And I'm not going to explain the exact nature of a pseudogene. I'll very simply, because you can look that up just about anywhere. But these 8 papers, 3 of them are published or 4 of them actually are published in Nature, by the way. And Nature and Science are the 2 effectively Bibles of modern biology. If you get published in Nature or Science, you are on track. That is high level quality research. Okay, so people are publishing in these journals showing that pseudogenes actually produce RNA that is very important in regulating the genes that they seem to be related to, that they seem to have been duplicated from. So it's very possible that they are not evolutionary leftovers at all. In fact, it's highly probable that they are necessary regulatory components that are either producing RNA that interferes with the production of a gene or transcription of a gene, or that enhances the transcription of a gene. So they're playing a role in that finely tuned back and forth of how much protein gets produced when and where. Okay. And if you think about this for a second, and the magnitude of this is really difficult to wrap your mind around if you haven't just been living it and breathing it. But you started out as a single cell, and you now have trillions of cells in your body, and all of those cells do vastly different things. And your eye cell and your skin cell and your liver cell and your intestinal cells all have the same information in them. Okay. So every cell is carrying around the entire library. But if every cell produced the same proteins in the same quantities at the same time, you would not be a human being. You would be a collection of amoeba. Okay. Because you would not be able to function as a multicellular organism. And your development too, it changes over time. When you were a single cell, you needed to produce different things in different ways than you do now that you're an adult and all of your tissue is differentiated. But when you're a child and you're going through puberty, you have changes in gene expression. When you're going through a growth spurt, before that, you have changes in gene expression. When your brain is developing all the way up to the time that you're 25, you have changes in gene expression. So you have this vast amount of information that needs to be precisely timed, not just across space and across different kinds of tissue, but across time, your whole life. And you have to be able to pass all that information on down to your offspring. So you have a small percentage of your genome that's genes, maybe 2%, but the other 98% tells you what to do with those genes and when and where. And that is super important. It is not useless information. And cutting edge science is telling us it is not useless information. So first point for evolution, most of the genome has to be useless. Boo, that doesn't work. Okay. Second point, most mutations have to be harmless. Now I'm going to do a little analogy here that's going to help get my point across. All right. Because evolutionists like to argue that if mutations are neutral, so that means they can't be selected against, that means that they don't actually have any effect on the genome whatsoever, and they're really no big deal, right? But I'm going to show you 2 neutral mutations in a row. And I think you'll conclude the same thing that I conclude, which is as I add neutral mutations, they don't stay quite so neutral. So if I texted you this message, this short message, you might think that I had attended Franciscan University of Steubenville, because they say that there a lot. If I texted you this short message, you might think that I don't proofread my text messages very well, and you'd be right. But if I texted you this short message, now we've departed from the meaning God is good, and you're beginning to wonder if I'm trying to explain to you in very broken English that I understood what you just texted me. Right? So we took two mutations that were neutral by themselves. They didn't alter the meaning of the sentence. We put them together and it completely altered the meaning of the sentence. Now, this is a little bit oversimplified because I'm only dealing with 12 characters, including spaces. All right. But this gives you an idea of what's happening in your genome. As you accumulate changes over time, negative things show up that didn't show up in a previous generation, and they couldn't be selected against. If you look at the beginning of population of giraffes under overproduction, there's a significant variety of coat color in that giraffe population. If you look under adaptation, after natural selection has had its way with the variation in the population, we have a much more uniform population. Natural selection eliminates genetic information. Okay, it doesn't help with variety. So mutations give us variety. Natural selection eliminates variety. It is supposed to eliminate all the bad varieties, right? But just to put a number on it, in order to eliminate all of the negative mutations, and these are just the negative mutations that are obvious. We're not even talking about the nearly neutral ones that I was just explaining and giving an analogy for. To eliminate all of the negative mutations that are obvious in a population, you would have to eliminate about 90% of human beings at every generation. So we can't actually weed out all of the super negative mutations, let alone all of the slightly negative mutations. It's impossible. Okay. And there's an additional point here that I wanted to bring up, and that's that fitness is not fungible. So when you have money, it's fungible, right? You can make a budget and you can say, well, I'm going to put $100 towards this and $400 towards groceries and $50 towards gas in my car. Well, if I don't spend $100 on groceries, I can spend that $100 on the gas in my car or on whatever the heck I want, movie tickets or something like that, right? Okay, so fitness doesn't transfer the way money in your bank account transfers. So if you have something that's damaged in one area and something that's beneficial in another area, that benefit might be good, but the damage that's accruing in the other area that goes along with the benefit is a big problem. And evolutionary biologists can't just whitewash this away. And I'm going to deal with this a lot when I deal with the next problem. Okay, so we've kind of established mutations can't really be harmless, but I'm going to go way into that in the next mutation myth, which is this idea that we have beneficial mutations. So if you read any scientific paper whatsoever that deals with genetics and evolution, you will see that mutations are overwhelmingly negative. Every abstract of every paper, every introduction starts with the idea that mutations are overwhelmingly negative. Most mutations are negative. Almost all mutations are negative. However you want to say it, they're almost all negative. Everybody knows this. This is actually not a debatable point. Okay. The point that's being debated is, can there be beneficial mutations that outweigh all these negative mutations? And if you're wondering what the graphic is with all the lines, this is actually a tracking of the different mutations in the COVID virus from December 2019 to April 2020. And do you see how most of those lines end before April 2020? That means that virus is no longer in circulation because the mutations that it sustained were negative. Okay, we see this happen in real time, all of the time. All right, so some objections that are raised to this idea that there aren't beneficial mutations are, well, wait a minute, antibiotic resistance is beneficial. So let's look at MRSA, right? Everybody's heard of MRSA. MRSA is methicillin-resistant Staphylococcus aureus, and it's a big problem in hospitals, right? It expresses a protein variant in the cell wall that methicillin can't bind to. So since methicillin can't bind to it, it can't break the cell wall, it can't kill the bacteria, right? But if you've ever done any carpentry, you know that something that's square works a lot better than something that's not square. Okay, so you can think of it as this protein being mutated causes a lack of squareness in the proteins that are cross-linked in the cell wall. So that actually overall decreases the integrity of the cell wall. And this is anecdotal, but a nurse that I know has shared with me that most nurses in the hospital system, they actually have MRSA growing in their nose because you have Staphylococcus aureus on your body. Okay, so they have MRSA growing on their body, and if they stop working in a hospital, within a very short period of time, they no longer have MRSA growing on their body because it's rapidly outcompeted by the normal non-resistant Staphylococcus aureus because there's just no way for the damaged cell wall of the MRSA to compete with the integrity of the cell wall of the non-resistant bacteria. So if you remove the antibiotic, boom, benefit's gone, and that strain is gone. It's just gone. It disappears from the population. Right. There's some other objections that are raised. I actually found this one in a video that was supposedly refuting genetic entropy. His examples were terrible. He said that tusklessness mutation in elephants is beneficial because, where there's poachers, they're not going to kill the elephants that don't have tusks. But he failed to mention that the tuskless mutation is connected to an X-linked lethal gene. Okay, so if you're inheriting the tuskless mutation, you're also inheriting this X-linked lethal gene. That means that only females are tuskless, so males can still be hunted to oblivion by poachers, which is actually really bad for your gene pool. It's extraordinarily bad for your gene pool, because if you take out most of the genetic variation in the males, you're going to have a very genetically homogeneous population, and then you're going to have a genome catastrophe, which is exactly what we're talking about in genetic entropy. And it's exactly what we see with every endangered species on the planet. They're just headed towards genomic meltdown. They're not going to be able to reproduce after a certain period of time. Okay. It also means that these tuskless females are only going to produce about 67% of the offspring as females that have tusks, because half the males they conceive are going to die in utero. Okay, so now we've taken a population that's already endangered and we've reduced its fertility by a third. You're just going to have extinction after a while. This is not a benefit. Okay, there's another objection, sickle cell trait. It offers protection against malaria. Well, it confers a slight increase in survival in individuals that are infected with malaria over individuals who have normal alleles and don't have sickle cell trait. But if you've got 2 copies of the sickle cell trait allele, you have a 90% chance of never reaching adulthood. That's a detriment. Okay, it's bad for the population. And even 1 copy, the copy that makes you resistant to malaria, can cause you all kinds of problems if you're doing heavy exercise or athletic training, if you're dehydrated, or if you're at high altitudes. This is not an example of a beneficial mutation. It's an example of how a negative mutation stays in a population because it comes with a little bit of a benefit under certain selective pressures. Okay, this is actually a negative mutation. Don't let people fool you otherwise. I know previously in the debate, lactase persistence was brought up. I just want to bring up 2 quick things about it that are actually important. And one is that being lactose tolerant actually has more to do with your microbiome than it has to do with your genetics. So there are people who do not have the appropriate mutation to metabolize lactose themselves, and their gut bacteria do it for them. So you don't actually need this mutation in order to get the benefit of being able to eat lactose. And the benefit of being able to eat lactose really is not actually that important to your survival. Because there's a lot of other things human beings can eat, and there are all kinds of populations that survive without milk at all. All right, the final objection that's been raised against genetic entropy on this particular point is that, well, eventually mutations will reach an equilibrium, okay? So as negative mutations accumulate, the argument goes, then beneficial mutations will be more likely. Eventually we'll get to a balance, okay? So you see things in scientific papers like this. This is from the Proceedings of the Royal Society in London. For example, the total of fitness degrading and fitness increasing effects that get fixed might be in equilibrium such that there's no unbounded change in fitness in most populations. This is historically how evolutionary theory has always progressed, because Darwin himself started out saying one might suppose, right? His whole theory was littered with one might suppose. One might suppose, or it might be, or it mathematically works out, is not a proper biological argument, okay? One experiment that shows that your mathematical model is incorrect completely destroys your mathematical model. All right. And according to reliable sources, there's somewhere between 7,000 and 10,000 human genetic disorders that are caused by mutations in single genes. Okay. If mutations reach an equilibrium, I want to know why, after 3.5 billion years of supposed evolution, Where are the 10,000 human genetic conditions that are beneficial that are caused by mutation in a single gene? We are not at mutation equilibrium. And if we're not at mutation equilibrium after 3.5 billion years, I don't know when we're going to get there. This is a spurious argument. It's done based on mathematical contingencies that don't take into account that after you reach a certain level of mutational load, you're just going to die. Okay, your proteins are not going to work. Your RNA is not going to be transcribed properly. You are just not going to be viable. Okay, this brings us to the question of new information. Evolutionary biologists like to argue that mutations can create new information. And in these two sentences, technically, the second sentence is new information. All right, but that's not the kind of new information that's going to be helpful for evolutionary theory. This is not even the kind of new information that's going to be helpful for evolutionary theory, because to go from the smallest genome of a free-living bacteria, which is represented by God is good, to the genome of a human being, which is represented by question 6 of the Summa Theologica by St. Thomas Aquinas, I would actually need 3 times this much additional information in order to go from a bacterium to a human being. So this isn't even a fair representation of how much new information I'm going to have to contribute. Yet we get examples like this all the time where evolutionists say, oh, look, there's new information. I'm sorry, it doesn't work that way. All right. The best example evolution has got, Dr. Ken Miller references this example, is Dr. Barry Hall and the, quote, evolution of the EBG gene. So what Hall did was he knocked out the ability to produce lactose in E. coli by deleting the LAC-Z gene. And he then grew the E. coli on lactose-only media. So this is stuff that only has lactose sugar in it in order for the bacterium to grow. And he found that mutants arose a lot faster than he expected. And this is really interesting because it's going to tell us something that the evolution that he's anticipating is not actually what's happening. And we'll see that here in a couple more steps. So the ability to metabolize lactose came from a mutated protein called EBGA. So Hall went back to look at the original unmutated protein, which he called EBGO. It turns out that this enzyme was able to break down lactose in the test tube. It just couldn't break it down fast enough for the organism to survive. Okay, but it already breaks down lactose. So Dr. Hall cited 21 other instances where bacteria supposedly evolved the ability to metabolize a new sugar. And of these, 9 of these mutants constitutively expressed an enzyme that they already possessed that had a slight affinity for the new sugar, just like the one that he supposedly, demonstrated evolved. Right? 4 of them were mutants that had gene duplications of enzymes, very similar to what I just described, or enzymes that were produced more abundantly, again, similar to what I just described. 1 mutant already possessed a second subunit of protein that could already do the same job as a knocked out protein. So it basically, like I said, had a redundant system. And this is common actually with bacteria for redundancy to happen and all these just in case genes in case, the environmental changes in one way or another. This is one reason that you can get these experiments that look like there's high rates of beneficial mutation in the laboratory when really there's not, because what's happening is these bacteria are shedding these just in case genes. So then they grow faster under laboratory conditions. But that doesn't mean that they've actually benefited anything. And they're certainly not on their way to evolving to anything more complicated because they're losing genetic information, not gaining it. Right. 4 of the mutants, quote unquote, gained the ability to bind to a new sugar by losing specificity for the original sugar they metabolized. Again, this is a big problem because if I lose specificity for my original substrate as an enzyme that gives you a leaky enzyme, that means the enzyme is going to be cutting things it shouldn't. And when you have little molecular scissors running around chopping up things that they shouldn't, it's kind of like having a toddler running around with scissors chopping up things that they shouldn't. Bad idea, bad for the cell. Okay, 2 mutants had enzymes present that could already metabolize the sugar, but they were now activated by the presence of a new sugar because of a loss of specificity in the activation mechanism. And 1 mutant was able to transport sugar into the cell when it previously could not because of a loss of specificity and transport, kind of like I talked about earlier with antibiotic mechanism. Okay, so that's a lot of examples that I've gone through in a very short period of time. Some of them are a little technical, and I'm sorry if that skipped across the surface a little bit. But the take-home point is that anytime an evolutionary biologist shows you a, quote, beneficial mutation, you need to ask them, what's the cost? And what happened with the original protein? Okay. Is it actually damaging to the fitness of the organism in some way that you're not telling me? And what happened to the original protein? Does the original protein have more specificity or less specificity? If it has less specificity, we're going down, not up. Okay. Dr. Hall himself stated at the end of a paper discussing the EBG mutation, and I'm going to read this without the technical jargon, so there'll be a couple of translated bits. A variety of new functions can arise from single point mutations within a gene. All of the new functions evolved for the EBG enzyme, however, represent variations upon the general function that it already had. The ancestral enzyme does not hydrolyze any of the sugars that we tested at biologically significant rates. However, it breaks all of those sugars down in the test tube if we use the purified enzyme. Thus, in a real sense, the point mutations that generate the evolved enzymes that we studied do not result in truly new activities. They simply improve old functions. We have no new information. We have no new functions. He just said it himself in a peer-reviewed article published in Evolutionary Biology. Okay, the last objection I think I'm going to have time to talk about is a compensatory mutation in a flu virus that someone published in Science. They described a mutation that rescued a lost function in the NA protein on the flu virus, and the flu went right back up to producing at the normal rate that it had before. Okay, so evolutionists want to trot this out and say, look at that. You can have compensatory mutations. So really, there's no problem with negative mutations. It doesn't really matter if they're harmful, right? Wrong. Okay. This mutation was engineered. It was specifically selected by the researchers, and it was artificially induced into the flu virus. And it's actually a mutation that wouldn't be as likely to happen on its own. Now, I took the data from this paper and I looked at the length of the gene in question, the relative likelihood of transition versus transversion mutation. So I kind of calculated it out, and to be as reasonable as I thought, because even though technically you could have 3 possible mutations at any locus in the genome, only one of them is super likely. The other 2 are half as likely. So technically, you really only got 2 mathematical possibilities, right? So the chance that rescue mutation would happen mathematically before another mutation further corrupts the gene, which then might not be rescuable, is 1 in 3.8 times 10 to the 550th. Now, for reference, there's only 1 times 10 to the 80th molecules in the entire universe. You're talking about multiplying the number of molecules and the number of molecules in the universe times the number of molecules in the universe times the number of molecules in the universe twice before we even approach the likelihood that we're talking about here. Like, you'd find 1 atom in the universe. This is mathematically impossible. Okay, so yeah, they engineered a back mutation that rescued this flu virus. That doesn't mean that it would happen in real life. Okay, the last problem is that the accumulation of mutations simply does not produce new kinds. We've witnessed this over and over and over and over again. The data that we have when we're speeding up mutations of bacteria, of fruit flies, of mice, it shows us we don't get anything different. We don't get a new species of bacteria. We don't get a new species of fruit fly. We just get strains of the same old species that we've already been mutating for over 100 years. And we've sped up that mutation up to 15,000 times. If there was going to be something new produced, we would have seen it. Okay. And even with species like finches that are supposed to have evolved all from a common ancestor, there supposedly are 14 different species of Darwin's finches. A lot of them can interbreed, such that actually at max, there's maybe 6. There's possibly only 1. Okay, we're not actually seeing speciation. We're seeing diversification in certain elements of external form, like beak size or coat color. We're not seeing new species. We're certainly not seeing new genus. Okay, if you think kingdom, phylum, class, order, family, genus, species, right? Species is just one kind of thing. A genus is a collection of a few species. We don't even see new genera. It's never been observed. Okay, we're not even talking about origin of families, let alone origin of phyla. Right? We just have oscillation around a mean. One of the most famous examples of this is work done by Peter and Rosemary Grant on the Galapagos finches. They looked at the finches during a drought and they said, hey, that beak size, that increased by about 5%. Look, evolution and progress. The beak's going to get bigger and bigger and bigger. And this is how this finch went from a little tiny beak to a great big beak. But you know what happened after the drought conditions were over and the rains came again? The beaks decreased in size by the same amount that they increased in size originally. There's genetic information that allows organisms to fluctuate around a mean and deal with changing environmental conditions. This is true in bacteria too. Even Pierre de Grasse, who is a prominent proponent of evolution, says, in some, the mutations of bacteria and viruses are merely hereditary fluctuations around a median position, a swing to the right, a swing to the left, but no final evolutionary effect. And why? Precisely because there's no room in the genome for the kinds of changes that evolutionists need to get from a microbe to a microbiologist. There's just no room for it. Everything is actually functional. And there's a lot of just-in-case stuff that allows an organism to adapt to certain changes within its environment, such that people can make preposterous claims like fruit flies evolve within a year. No, they from generation to generation, they change to adapt to the season. But if they were evolving directionally, by the time you got back around to spring, you'd have dead fruit flies because they'd be adapted to fall. They're just varying around their means. Okay, so let's conclude. We have a very good case for genetic entropy, right? Virtually all of the genome is now known to be functional. And the much touted claim of junk DNA is absolutely false. There's no truly neutral mutations because all mutations destroy information at the genetic level. And there's a great book on this called Junk DNA by Dr. Jonathan Wells. I highly recommend you check it out if you're super interested in the science behind this. He does a really good job of explaining a lot about why there can't be junk DNA and a lot of the genome is useful and these mutations are destroying information left, right, and center. Even the supposed beneficial mutations come with huge costs. And no matter how many mutations an organism accumulates, it still doesn't become another kind of organism. Okay, a snow leopard couldn't even give rise to an ocelot. That's how little genetic variation is actually possible. Okay, within the limits that we've actually observed. So to sum it up, if the genome is actually degenerating, it's bad news for the long-term future of the human race. It is also bad for evolutionary theory. If mutation and selection cannot preserve the information already within the genome, it is difficult to imagine how it could have created all that information in the first place. We cannot rationally speak of genome building when there is a net loss of information every generation. Dr. John Sanford, thank you very much for your time, and I hope that you learned something.